Good morning, everyone. Welcome to Breakfast Club, for which we are also welcoming two really special guests, one of whom you've seen before, but are, I know, excited to see back. We have our herpetology curator, Dr. Raina Bell, and our herpetology collections manager, Lauren Scheinberg. Hi, both.
Hello.
Yeah, and you all would not believe how much work I had to do to get Lauren to agree to appear on the show. She fought me for like 3 months, so this is a real accomplishment for all of us. And before I get into the show itself, I just wanted to take a second to acknowledge that Californians are being really, really hard hit right now in all the ways the rest of the world is, and especially with the wildfires that are happening. So just wanted to say that our thoughts are with absolutely everybody impacted by those, and our gratitude with everybody who's responding to them in any ways. And yeah, thank you for being here today. And I'll dive in by saying or asking each of you to tell us what a curator and collections manager does, respectively. And
oh no, Laurel. I think Laurel just froze. Okay, Lauren, do you want to go first and say what a collection manager does?
I'm Lauren, and I'm the collection manager for the herpetology department here at the California Academy of Sciences. That means I am basically a librarian of our research collections of dead and preserved amphibians and reptiles. So in our collections, we have about 320,000 specimens, frogs, salamanders, snakes, lizards, crocodilians, turtles. And I function like they're kind of the keeper. So I send them out on loan, I receive them, I catalog them, I put them on a database that's accessible to everybody online, and I help researchers, students, scientists use our collection for their own work.
Excellent. Sorry, I disappeared there for a second. Raina, will you tell us what a curator does? And I'm going to awkwardly switch computers here, so sorry if I disappear for a sec.
No worries. Yeah, so a curator has sort of more of a advisory role in the growth and use of a scientific research collection at a natural history museum. So Lauren oversees the day-to-day operations and safety of this really important resource for scientific research and conservation. And I help with sort of big scale decisions like what are groups of organisms that we don't have very good representation of in the collection, and how can we improve the way that the collection is being used by researchers. And then I use the collection for my own research as well and help contribute to its growth.
Excellent. Thank you so much. And you guys have spent you two have spent the last handful, sorry, I had to change that. My mom gets really mad. You two have spent the last handful of weeks intensely debating how to kind of curate a even tinier collection within the collections that show us today, which is a lineup of 10 specimens. Do we do 10? Okay.
We fudged the numbers a little.
Right. I mean, on which side, lower or higher?
It's higher, but like they're kind of grouped together.
Yeah, no, I approve. I love this.
Yeah. We have one of the most significant research collections in the world for herpetology. So it's pretty hard to narrow it down to just 10 things to show off just, the cool things, but also the things that are really significant for research. So
yeah,
we're the 6th biggest in the world right now. And we've been growing our collections from like the mid 1800s until now, and we're still growing the collections. Unfortunately, in the big 1906 earthquake and fire, we lost a lot of collections from before that date, but we also adopt orphaned collections. So a lot of smaller natural history museums or even bigger natural history museums sometimes can't support their collections, and so we'll take them in. So we did that with the Stanford collection back in the 1950s, 60s, and that now accounts for our oldest collections.
Neat.
So we still do have stuff from pre-1906.
Yeah, that's amazing. And I'll say to viewers too, before we move into starting to show you some specimens, you're welcome to ask questions anytime. We'll kind of incorporate them into as Raina and Lauren are talking, and then we'll ask a handful more at the end. So please feel free. Comment section on Facebook and chat box on YouTube. And what's most helpful for getting started? Should I give you your slides, or you tell me?
I think I'll say one thing right before the slides, because this one specimen isn't in the slides. So I'll start off just like a random thing. So in the collections, we preserve things usually in 3 different ways. Most of the specimens you'll see today are wet specimens, and so they're in alcohol, either fixed with alcohol or formalin preserved, kind of like embalming the body, but ultimately stored in 70% like drinking alcohol. Another way that we preserve specimens is skeletal material. And so this is an articulated snake in like a archival deli container. But some of our specimens, especially ones that aren't in great condition or maybe don't have the best associated data, will skeletonize like this. And then a third way I wanted to show, which is one of my favorites, is cleared and stained. So we don't do this as much anymore, but this was a way of preserving a specimen where you take off the skin and then you use a variety of chemicals and heat to clear the flesh. And so all of the muscles are still there, they're just clear. And then we use 2 different dyes. One dyes the bones red and one dyes the cartilage blue. So it's a really nice way to see all of the internal structures and all of the bones without it kind of falling apart, which sometimes it'll do if you just preserve it as a skeleton. So I don't have a slide for this, but I just really like to show it off because they're really cool ways and good looking ways to preserve specimens. They're a little less common now because we use a lot of CT scanning to look at internal structures, which we can talk about a little later.
Great. And sorry, did you say, was that was the clearing and staining kind of an older technique, or when was that started? When did that start being used?
It's a little bit older, but it still is used now because sometimes, especially with like tadpoles and things that are more like cartilaginous, it's easier to see some of the structures inside that you wouldn't otherwise, but it's less common now than it was.
Great. Yeah, and really beautiful.
It's a little destructive. So if you can look at the internal structures now through other means without having to, I mean, in some ways destroy the specimen, then that's kind of preferred.
Great.
Yeah.
It's kind of a dying art form too. Like it's not something that many young herpetologists or vertebrate biologists are trained in anymore necessarily. So yeah, it probably won't really be practiced that much moving forward just because people aren't learning this skill anymore.
Is it something that we do at the Academy?
It's done more in ichthyology. So in the fish department, they still do quite a bit of it. We have an ancient like oven in the lab that could be used for it. Some methods don't require the oven anymore. And we've had some researchers come through and clear and stain some specimens, but generally we don't do it. I don't know how to do it.
I don't know how to do it either. So,
okay, great. That was really beautiful, and thank you for showing us. So tell me what's next, or what would you would if you would like those slides now, or what would be helpful?
Yeah, let's go to the slides.
Okay, there you go.
That's us.
That's us and our colleague Erica, who is also part of our herpetology department. And since we can't actually be in the collection space today, like some of the other behind the scenes tours have been able to do, we did want to show you at least what it looks like, where all these specimens are stored. And so I think Lauren can probably best explain the sort of nitty-gritty of this space and why it's appropriate for storing specimens long-term.
So behind the offices and public floor at the Academy, on every floor we have these collection rooms. And so the one that we're showing here is our main wet collection room. And it's a fireproof, hopefully fireproof room filled with jars of alcohol. And all of the jars are on these compactors, and so these moving shelves. And so we can fit more shelves in. And I think each one weighs about 6,000 pounds, but it's on a crank system, so we can just crank them over and move them. We have jars that range from about 8 ounces in the collection to gallons. And everything that doesn't fit in a gallon jar goes in large metal tanks. So we have a room full of these coffin-like steel tanks.
And Rachel just asked a question about whether there are earthquake protections in place and what those are.
Yeah, you can see on the shelves there's a little rim, and those are earthquake bars. And then we have a fire suppression system inside the room that gives off this like high density mist that is supposed to protect the collections in case of fire.
Mm-hmm. Cool. I like that also when you put that photo up, you just gave people a few seconds to really like take in the splendor.
Totally planned. And then, so most of our collections, as Lauren mentioned, are whole animals preserved in ethanol, which is very flammable, obviously. And so that's why we have this very sophisticated facility for storage. And then part of the reason that we aren't in the collection itself today is because we do have other preparation types that are in different rooms. And so it would have just been very logistically complicated for Lauren to move between them to show you guys things. But I think she can still tell you what those other rooms are and what's in them.
Yeah, so just to say, in this building, we have an amazing amount of doors. So to get into the collection, I would have to go through like 7 doors holding a camera. But we have 2 wet rooms or wet collection rooms filled with jars of ethanol. We have one of those is shared with entomology, so insects, mainly arachnids, and it's also shared with birds and mammals. Both of those collections are primarily dry. They have beautiful dried specimens in other collection rooms, but they have a small collection of alcohol preserved specimens, which are in this room that you see here. We also have the tank room I mentioned, which we share mainly with ichthyology, or fish. They have a lot of big fish in big tanks. We have another dry skeletal room. So all of like that articulated snake skeleton that you saw, that is stored in another room in big cabinets with other dried fish skeletons. And then we have a tortoise room, which is an oversized collection that's mainly filled with Galapagos tortoises and sea turtles. We'll talk a little bit more about our Galapagos collections later in the talk, but we have the largest collection of Galapagos tortoises in the world, and they take up a lot of space. And then,
no, I was going to ask, and they're very old. Is that right?
They're very old. Most of them are from 1905 and 1906. Mm-hmm. We have a couple that are a little older and a couple that are a little newer, but the majority are from a single expedition to the Galapagos.
Right. And Krishnan asked a question that is probably on a lot of people's minds, which is, are visitors allowed in these rooms? And I think so I know that visitors can get a collections tour with some of our behind the scenes tours when the Academy is actually open. But other than that, other than those kind of guided tours, which are, we do want the public to see this, but obviously the holdings themselves are so often fragile and valuable, and in many cases old. How I know that specimens are loaned out to other scientists doing research, but maybe you could just kind of talk about how they circulate a little more, how people do see them.
For the most part, we don't do public tours. Occasionally, quite often I'll do tours for educational groups or mainly educational groups. And then we have, as we mentioned, the behind the scenes tours, which are organized through the Academy that kind of dip into a bunch of different collection rooms. But for the most part, these aren't for public display or viewing. All of the information about the specimens is online, but it's a rarity that you get to go and visit in the collections.
Right.
Yeah, so we host mostly research visitors, and sometimes artists or other people who are interested in illustration or accessing specimens for other still educational purposes will visit the department, and Lauren will go into the collections and bring out the specimens that they're interested in looking at more closely. But for the most part, this is an active research collection. So it's kind of like a rare book library where, like, yes, everybody wants to see the rare books, but taking them out of their specialized storage is a little bit risky and potentially damaging. And so we really only want to bring them out for a specific research purpose, and for people who know how to handle them carefully and who are going to be taking important scientific data from them.
Right. And that's why we do so many programs like this too, because we do want the public to be familiar with how these are used and that they are there. But but risky, like you said, and also logistically tough sometimes.
But we do function as a library for scientific research. And so I send out, in a normal year, I would send out about 1,200 specimens and 1,200 associated tissues to researchers around the world. And so when I do that, I take whatever specimens I have that they want out of the jars, I wrap them in alcohol-soaked cheesecloth, and then I triple bag them and send them in the mail. So they're sent like that all around the world for researchers to use.
Excellent. And I just saw a comment from Mo Flannery. So this is our mammalogy and ornithology collections manager watching, and she has committed to also coming on this show. So Mo, we're holding you to it. Yeah. And I'd love to see what you pulled today if you're ready for that.
Yeah, let's do it. Let's move on.
Okay. So the first set of specimens is an international face-off for cutest desert lizard.
Oof.
And so on the left, we have the horned lizard, Phrynosoma platyrhinos, which is a species from the desert southwest of the United States. And on the right, we have the thorny devil, which is also a desert species, but from the deserts of Australia. And so, as you can see, they look pretty similar. They have similar cute little smushed in faces and spines all over their heads. But these are actually not closely related species at all. They're in completely different families of lizards, but they look very similar. And so Lauren can show you some more of those features, and then we'll also tell you a little bit about some of the other adaptations that they have for living in the desert.
So I'm going to start with Moloch. This is Moloch horridus, also known as the thorny devil. It was named, the name Moloch comes from this biblical god that is said it was like the god of child sacrifice, but this is Moloch. It's very spiky. This one is from Western Australia, and it was collected in 2013. But you can see you wouldn't want to step on it or try to eat it.
Yeah. Yeah, and they're just like totally insane looking lizards.
And it's got like a little tiny face. Both of these lizards mainly eat ants.
Really?
Yeah. They have really similar lifestyles on opposite ends of the earth.
Mm-hmm. And how does the spikiness help? Do you know?
Well, I think it prevents things from eating it.
Yeah, so eating ants is not super nutritious. So you have to eat a lot of them. Also, ants are small. Mm-hmm. So they have to spend a lot of time looking for food. And even once they find food, so like an ant hill, they have to spend a lot of time sitting there, slowly eating ants one at a time. So they're exposed to potential predators for a really long time while they're feeding, and they have to do that every day.
Mm-hmm. So much different than like an animal with a hunting strategy where they're underground and just kind of like lashing out once in a while.
Exactly.
since both of these animals live in kind of rocky desert habitats, they use kind of a camouflage technique as well to protect themselves. So they can both hunker down, close their eyes, and pretend that they're rocks. The other one that I have is Phrynosoma, also called a horny toad, but it's not a toad, it's a lizard. So we prefer the horned lizard. This species this one was collected in Nevada. I collected this one. But once again, yeah, it's an ant eater, sits out in the desert. They're not very fast moving. I'm not supposed to say it, but they're pretty cute. One thing that both of these species can do as an adaptation to the desert environment is they both use capillary action to absorb water from the environment. And so since they live in really dry areas, any atmospheric moisture, they can kind of use their ridges on their skin to collect it and then use that water.
Yeah, it just sort of like funnels to their mouth.
Yeah.
Wow.
Which
Yeah.
is pretty amazing.
Seriously.
And also really cool research that people have figured out in these, again, two very distantly related species on opposite ends of the world, but that are adapting to a really harsh environment.
Yeah. I also feel like we should just give ourselves a pass for the rest of this stream and not judge or punish ourselves for thinking things are really cute or really adorable.
It's going to happen again.
Yeah.
So.
When you put those tiny faces that close to the camera, it's impossible not to make embarrassing noises.
So,
Yeah.
but there, in my mind, there's one winner. And that is, I don't need the big screen for this because I'm not showing off anything. It's the next slide is.
Yeah.
Okay.
So we have a nice video to demonstrate why the Phrynosoma or the horned lizard is supreme, even if its name is maybe a little bit more humble than the thorny devil. And that is because some, but not all species of horned lizard can shoot blood out of their eyes at potential predators. So we're going to play this video for you. Oh no, there's an ad.
And
Wow.
I think the action is it's far in the video.
Yeah, but it should start, it should be queued up to play like partway through. If not, I'll advance it. Yeah, there we go. So here it is, just minding its own business, eating ants. Just a regular day in the life of a horned lizard. And this is a potential predator. And they can run, but they're not super fast, like Lauren said.
Usually if you encounter them, the first thing they'll try and do is hunker down and pretend that they're not there. And then when they realize that they can't do that, either squirt blood or run.
Oh
Yeah.
that's such amazing volume. Like and
Yeah.
So not only is it just blood, but because they primarily eat ants, which are acidic by nature, they can use some of that acid. And so the blood that they squirt out is actually acidic, nasty,
oh wow.
bad tasting blood.
And that's why that coyote reacted so was it coyote?
Yeah.
Reacted so dramatically to that.
I mean, also, it would be pretty startling. Regardless of the taste or scent of the blood being squirted in the face with
Fair.
blood, probably a little upsetting.
Yeah.
I just want to note, though, that it is really stressful for the lizards to squirt blood out of their eyes. And so if you find them, don't try and provoke them to the point where they have to do so.
Right.
Yes.
I think energetically, it's not ideal for them.
Yeah.
And also, if you're in a part of the state of California that has horned lizards, you should be keeping an eye on your dogs and keeping them on a leash, most likely.
Mm-hmm. The anatomy of like the musculature involved with squeezing out that at that velocity must be, I mean, is it when you look at it, is it noticeably different than other lizards?
That's a really good question. I don't know if anyone has gone into detail on the musculature of the eye, but it would be a really good group to study for that because it's not just like a one-off where only one species does it, but you have a little bit of variation where some species do and some species don't. So yeah. I wonder if someone does
it. About half can stoop blood.
That's really well. Maybe there's some younger person out there right now who just suddenly identified a new career path. I hope so. Let us know what you find.
But
somebody should study this.
A lot of these lizards are also protected because they were really big, I think, in the pet trade, even though they don't do very well in captivity. And so, for the most part, you're not really supposed to harass them or anything.
And Candace asks, she says, there are horny toads in the San Joaquin Valley. Would they be the same species as what you showed?
No, they're not the same species. In the San Joaquin Valley, they're the coast horned lizard. And these ones that I showed are further like Great Basin desert species. So we do have a coast horned lizard here that you can find like in San Jose and like Henrico State Park, and then all through the Central Valley hills.
And generally they're close relatives. Like all things that we call horny toads are the same as horned lizards. It's just a different terminology. And like Lauren said, we prefer horned lizard just because it's a little more correct in terms of the type of animal that it is.
Well, props for Candace for putting toads in quotation marks.
Yeah.
Oh, yeah, totally. That was not at all meant to be a criticism.
No, I know.
But yeah, so there are quite a few different species, and you can find different ones in different parts of the Southwest.
Yeah. Oh, excellent. People are loving this, and the video in particular, not surprisingly.
It's a good one. But yeah, as Lauren said, these are a little tricky to keep in captivity, in part because of their diet, because you would just have to have so many ants on hand to keep them fed. And so that does make it a little bit harder to study them in a lab setting, which might be why this blood squirting behavior isn't super well understood.
Right. Okay, great. Can't wait to see what's next.
Should I do the next slide?
Are you showing? Oh, I forgot.
Oh, I was going to do next slide. I didn't know.
Next slide. Okay.
Sorry. Okay. So the next thing that we're going to show you are a couple of specimens that are related to some research that I'm working on with colleagues at the Natural History Museum in London, at the Smithsonian Institution, and the University of Texas at Arlington. So we have a big project where we're studying the evolution of vision in frogs. And I've talked about it a little bit in a night school program about a month ago, I think. And so one of the things that we're looking at is the evolution of eye size in different frogs and toads. And it varies pretty dramatically. And so Lauren's going to show you some specimens on extreme ends of the eye size spectrum, but I just wanted to give you kind of a photo preview just because it's a little hard to see the eyes through the internet. But so on the small eyes extreme end of the spectrum, we have the Mexican burrowing toad, Rhinophrynus dorsalis. And this is a species that does get into into the southern parts of the United States, but it's more of a Central American and Mexican species. So they have this very crazy body shape for a frog and very tiny little eyes. And that's because they have a burrowing lifestyle. So they spend a lot of their lives underground and they're not really relying on vision too heavily. Lauren is demonstrating some structures on the foot that help them dig in the soil.
Yeah, this little like spade-like structure here helps them dig into the ground.
Wow.
So you'll find that on a lot of burrowing frogs.
Okay. Yeah, so because they're spending a lot of their lives underground, they're not really using their eyes too much to sense their environment. And so we think that's why they have smaller eyes, kind of makes sense. And then on the opposite end of the spectrum, we have the Principe giant tree frog, Leptopelis palmatus, and like many tree frogs that you're probably familiar with, this is a species that has really big eyes. And tree frogs are often active at night. in really dim light environments, but they're still using their eyes and vision to sense their environment. And so they have really big eyes because it increases the amount of light that enters the eye and that the retina can detect. So it's kind of interesting because you have two different types of frogs that are both living in pretty dim, low light environments, but one of them isn't really using its visual senses very much, so it has very small eyes. And then the other end of the spectrum, they are relying really heavily on vision. And so to compensate for living in a really dim environment, they have really giant eyes. And so we're looking at eye size diversity across hundreds of different species of frogs to better understand how the lifestyle of the frog correlates with its eye size.
And that
And
frog,
we're using museum specimens to do that work, just because otherwise we'd have to travel all over the world, which is obviously not possible now, but even under normal circumstances, it could be really challenging to have to visit all these different parts of the world and find these different frog species. So having museum collections was critical to being able to do this very sort of basic first pass of what is the diversity of eye size in frogs.
and I was just the Principe frog was so much larger than I expected from the.
Oh yeah, maybe Lauren can show that again because it's a really cool species. It's one of the biggest tree frog species in the world.
So just as a note about wet collections is for the most part, color isn't preserved in alcohol.
Right.
This species comes in kind of a variety of colors, and this one that actually Raina and I found together was way darker, and it was pretty much black. But this species in general ranges from kind of a greenish to like a speckly black and yellow to a black.
Mm-hmm.
But yeah, this is a very large tree frog. You can tell it has these enlarged toe pads, which are good for climbing and holding on to things. Yeah, but this is an especially large one.
And it made me, I want to say too that Raina will be back next month, I think in mid-September, to talk about the biodiversity on Principe and the surrounding islands too. So you'll hear more about that frog, I hope.
Oh yes, for sure.
Very
yeah, and I just have here kind of a zoomed in, again, showing off the cute faces of the specimens, but here you can really see how just proportionally the eyes of these two different kinds of frogs are magnitudes different. And we find that it does really reflect the differences in their lifestyles, which makes sense.
Yeah.
So we did want to show another amazing video of the burrowing frogs. And that's just to show you that they do come above ground occasionally. And when they do that, it's pretty amazing. So we'll see. Hopefully this won't have an ad, but if it does,
we'll start charging them for it.
It's very quiet, but it's a bizarre noise normally.
I kind of love it.
Yeah, so that's the sound of a breeding aggregation of these burrowing frogs.
And where were they native to? I'm sorry to interrupt.
Sorry, what was that?
Where are they native to?
They're native to southern North America. They go into Central America. So you could, if you get very lucky, you could see this in very southern parts of the United States that are on the border with Mexico. But you have to time it just right, because these types of frogs that spend a lot of their lives underground are really only coming above ground to mate for the most part. And they're doing that after a big rainstorm when there's water available for them to mate in and for their young to develop in. So you have to time it just right, but also they make a ton of noise. So that does help you zero in on where you should go to find them.
That's excellent. And we'll also, I was going to say, we'll drop a link to the Nightlife episode that you mentioned in the comment section so people can watch that too.
Oh, great. Yeah, there were some other great presentations about frogs as well for frog fans.
Yeah.
Okay, so our next specimen is also kind of a famous one. So this is Conrawa Goliath, or the Goliath frog, which is found in Cameroon, and it also gets into Equatorial Guinea in Western Central Africa. And these are the largest currently living frogs. There are some frogs in the fossil record that are bigger than this, but these are enormous frogs that you can still see today. And this child here is holding an individual that weighs about 2 kilograms, but they can get up to 4 kilograms.
Wow. And I see Lauren putting into a tub.
Lauren's preparing to show you the real deal.
So as I said, our biggest jar is a gallon, and anything that doesn't fit into a gallon goes into a tank. And so most of our Conrawa specimens are in tanks. And so this is one of our tank specimens.
Wow, it's kind of roughly the same size of you.
Well, further back, and it's
Perspective.
bigger.
Wow.
But yeah, it's a large frog. Unfortunately, some of ours don't have associated data. So for the most part, in our collections, we only accept specimens that have data. So where they were collected, who collected them, identifications, and things that were collected legally unless they were confiscated by officials. And so a lot of these Conrawa that we have were confiscated by Fish and Wildlife or other agencies. I think most of the ones that we have were imported for the Calaveras and other jumping frog contests. So Mark Twain wrote a book about it, but they used to have these big jumping frog contests. I think they still do, actually. And people thought that these huge frogs would do really well in the jumping contest, but the truth is they don't. So they imported a bunch to try and make them jump, but these are fast-moving water frogs and really aren't great at jumping. But a lot of them were confiscated, including this big one we have here.
Wow. And what is their life and diet and everything else like that justifies that kind of size?
So it's remarkably poorly understood. And in general, just why frogs are big or small is something that's still not, in general, different species, why it's still not really understood what the selective pressures are. This species is heavily hunted as a source of protein. And so there is a lot of interest in understanding their ecology better, just to better communicate about their conservation and to make sure that they don't get overexploited. Mm-hmm. And so in doing some of that work, there's been some really interesting research coming out of Cameroon on their mating behaviors and where they lay their eggs and what their nests look like. So it looks like they build nests in the riverbank, and that to do that, sometimes they're moving around like pretty big size rocks to basically make like a mini little hot tub for their babies to develop in. And then they do also guard the nest. And I don't know if they move or sort of expand the nest to keep it from drying out, because that is one potential risk if you make a smaller body of water, is that it might dry out before your young have developed. But so they do have a lot of parental care and nest building that maybe they benefit from being a little bigger to be able to make those nests. But in terms of why they're so giant overall, still unclear.
But there are other species.
Their close relatives are like big-ish for frogs, but not nearly as big.
Okay, that's so interesting. We have a question from Natalia, age 10, who says, I noticed the specimens are stiff. Do they stiffen while they're being preserved?
They do, and that's one of the reasons why we preserve them is they'll last for hundreds of years, but they also stiffen up so that we can use them for research and they don't kind of fall apart.
Okay.
Yeah.
Thanks. Okay, let's see. Should I throw your slides back up?
Let's see what's next.
Sure, yeah, we're ready for the next one. So the next specimens that we're going to show you are the third type of amphibian that a lot of people haven't heard of before because they're mostly found in the tropics. And so those are caecilians. They don't have limbs and they have very secretive lifestyles. Most of the species burrow underground, and there are some species that are aquatic. And so here is Marcel, who is a PhD candidate at the University of Florida now, but he did his master's research at San Francisco State and was based at the California Academy of Sciences. And he's one of the few people in the world who is an expert in caecilians because, again, they have this very secretive lifestyle. Most people haven't even heard of them before. They're a little strange looking, as Lauren and I will show you in a second. And Marcel is a huge fan, which is amazing. And so he's doing a lot of really interesting research to understand their ecology better because it's just very, very poorly understood. And so his master's research, he was looking at two different species from Cameroon that live in the same habitats, but they look different. So the top species, Geotripides, has pretty well-developed eyes. And then the second species, Herpele, has its eyes are covered by skin and bone, so they don't have visible eyes. So they have very different sort of ways of sensing their environments. And it wasn't clear if they were using the food resources in their shared environment differently as well, and whether they have different hunting strategies, and whether they're found sort of at different depths of the soil potentially. And maybe that Geotripides would be more active on the surface because it still retains eyes, and maybe Herpele isn't. And so Marcel's research was looking at the differences in their diets using specimens. So here he is unpacking a bunch of specimens back at the Academy. And just as a quick summary of his research, he found that, yes, they do eat different things, which makes a lot of sense. So the Geotripides mostly eat worms, and the Herpele mostly eat little like hard-bodied crickets, little arthropods.
This is the Herpele. It's not that glamorous, but it looks like a worm. It's not a worm. It has a backbone. It's an amphibian. As you can kind of see, you can't see any eyes because, as Raina said, they're pretty much useless as they live underground. I did bring a second type of caecilian that's much larger just to show off what they can also look like. So this one is Dermophis mexicana. So it's from Chiapas, Mexico, and it's much bigger. And you can see its little face there. So this is a much bigger caecilian species. Some species, as a form of parental care, the babies will eat skin off of the mother, kind of like a form of nursing. Mm-hmm. So it's a fun caecilian fact.
Yeah.
But yeah, in general, there's still a ton to learn about their biology. There's only a couple hundred species that have been discovered so far, although there have been some really huge discoveries, like a whole new family of caecilian was discovered somewhat recently. And of course, just like everything else, when you just describe a new species as caecilian for the first time, usually you don't know anything about its ecology, its behavior, or any of its conservation threats that it might be facing. So there's still a ton to learn about caecilians, and it's awesome that there are people like Marcel who are dedicated to understanding them better.
Yeah, that's a very cool thing to be an expert in, and I hope
It's super cool. And also, like, there's very few people. It's a very small club.
Yeah.
But they are actively recruiting caecilian enthusiasts.
Yeah, make them more today. Also, good job, Emily Borger, who just made a really good Princess Bride joke about caecilians, which is, I heard you should never mess with one when death is on the line. So
nice.
Well done. Those are so. Yeah, I didn't expect the difference in size to be that, or I didn't expect them to be that different in size. That's interesting.
There's some really, really big species. Some of the aquatic ones get really, really big.
Oh, really? Like how big?
Oh, I mean, more than 3 feet, I want to say, like quite large.
Wow. That
I
definitely
think.
jumped from,
I've never seen one that big, but
yeah, that jumped from cute to horrifying like that. So cool. Okay,
Who's next?
let's see.
Good question. Oh, okay. I think we should actually start with the specimen for this one.
Okay.
As I was saying about data, is we try and get as much data as we can from each of these specimens. And so that includes the data that we take when they're collected. One thing to know about snakes is a lot of the snakes that we get were found dead on the roads. And so on a hot night, you can often go out and see snakes trying to absorb the last amount of heat from the pavement. And so it's a good way to see snakes, but it's also a dangerous place for snakes to be. So a lot of the snakes that we receive were hit. But we do get all of the data from them. We preserve them. We take tissues for DNA work. And then occasionally, we will look at their stomach contents. And so snakes swallow their prey whole and then slowly digest their food. And so sometimes, this is going to be a rough one to take out, but this is a rattlesnake. You can see its little rattle here, but then we also have its stomach contents, which I'll just show in the jar because it's going to be too hard to get out. You can see this is what came from its stomach, and it is an almost complete collared lizard.
Wow.
And it is roughly the same size, at least not the length, but in width of the rattlesnake itself. So we'll often take out, or researchers will take out the stomach contents to see what they were eating when they died. And then in another example, we have another rattlesnake, a local rattlesnake, with more digested stomach contents. And so in here is just kind of some digested fluff from, I think, a rodent. But it's important that we learn as much as we can from each of these specimens.
Very cool.
So, yeah. And so, as you may or may not know, snakes don't eat super often. And observing snake predation events, you have to get lucky, or you just have to dedicate a lot of time to it. And so using museum specimens has been a really important way for herpetologists to learn about the diets of snakes and how they change throughout the different life stages within a species, how different species that are living in similar habitats eat different prey items. So kind of similar to Marcel's research with the caecilians. And then also things like the relative size of a meal relative to the size of the snake predator. So all kinds of really interesting research that has come out of using museum specimens. And like Lauren said, really just trying to get like the most information that we can out of each individual specimen that we have. And this is just a really cool video that's sort of the opposite extreme of snake diet research, which is some researchers from Southern California who have been studying interactions between rattlesnakes and the different species of kangaroo rats that they predate. And they have this really cool setup where they have infrared high-speed videos. And so they go and find snakes that are out at night in an attack posture, waiting for prey items to come by. And then they set up their cameras and see what happens. So there's a whole series of these if people want to watch more. And this is just a short one to give you the idea of how amazing these interactions are.
Whoa.
Yeah. So, we think of like the rattlesnakes as being these really amazing predators, and of course they are. And this is just to show that even with all their specializations for being really amazing predators of small rodents, that it's actually a really challenging thing to pull off. And like I said, there's tons of videos of this, and sometimes the rattlesnake is successful, but I already really had an appreciation for kangaroo rats, and these videos just reinforce how amazing they are at evading these really fast strikes from rattlesnakes.
That was amazing. It's also funny how you can take this like super intimidating predator, and then when it whiffs that hard, it's just immediately funny. And you're just like,
It's a little bit
rattlesnakes. Yeah, that was incredible. Really, really cool. Neat. Yeah. Let's see. We have some good questions coming in, but I'm going to save the broader ones for the end. So, but you're welcome to keep leaving them wherever you're watching. Oh, okay.
So
do you have this poll?
now that we're talking about snakes, we realize that when people think about snakes, they often think about rattlesnakes, and then at the other extreme, these really large-bodied boas and pythons. And you might not realize that we also have boas that are local to California and to the Western United States. And they are very small and gentle in comparison to what you might initially think. So we have two species, the rubber boa and the rosy boa. And I don't know which one Lauren wants to show you first.
I have a rubber boa. I don't have
Okay.
a rosy boa specimen out, but I have a rubber boa because I think that they're great. They're a local species. I can find them like in the Bay Area. They're docile. They're not flashy at all. And they're wrinkly and brown and kind of cute. This is a rubber boa face and body. They mainly eat pinky mice, like rodents. And so their tail is really stumpy, and and it kind of resembles a head. So they'll use that as a little decoy. And so often they'll have really scarred tails from being attacked at their tails. But in general, they're smooth, brown, wrinkly, local friends.
Yeah, and one of the things that's really neat about boas, and we couldn't find a good photo of this, we were looking, but they kind of like a whale retains parts of the pelvis from when whales were hippo-like mammals living on land. Boas retain vestigial hind limbs as well, and they're just like a really tiny femur and a really tiny pelvis that aren't connected to the spine anymore, and they're positioned on either side of the cloaca, which is the opening through which everything happens at the back end of a snake. And so there's one on either side, and they tend to be a little bit bigger in males, and they use them in courtship. So I don't know if it's possible to see them on the specimen. They're really, really tiny, but it's kind of a cool thing where it kind of like a whale retains this vestigial structure that this sort of older lineage of snakes also kind of has a hint to their ancestry of having limbs.
Yeah, that's
and you can't really see it on this one, so I don't think it's even worth it to show it off.
That's okay.
Yeah.
Everybody is still, as soon as you showed the picture of the child sitting on the giant boa, you're bringing that out next. So this part's going to be disappointing no matter
I'm
how.
not bringing out the big one.
Yeah, sorry.
Yeah.
And then, so the other species, the rosy boa, is also just this like really gentle, docile, beautiful snake. They have these 3 lines down the back, and there's some variation in the color, but they're kind of like silvery pink and just this really striking, beautiful snake. And that one occurs a little bit further south.
Mm-hmm. Yeah, it's funny to think that you just think of boas as jungle snakes.
Totally.
Yeah. Neat.
So
all right,
we have boas too.
Boas. Yeah. Excellent.
All right, so this is kind of just good trivia prep, and also potentially useful if you're out and about and wondering, am I looking at a lizard or am I looking at a snake? Because although snakes don't have limbs. There are lizard species that also don't have limbs. And so snakes are kind of the most extreme version of a really specialized lizard that doesn't have limbs, but there are lots of lizards that also look superficially very similar. And so
locally as well. This one that I'm showing is not a local one though. So I believe this is from Armenia, but this is the European glass lizard or legless lizard. And it has no legs, but you can see, I mean, at least to me, it has a very lizard-like face.
Yeah, and you can clearly see there's a little dimple sort of at the edge of the mouth, and that's the ear opening.
Oh, wow.
Yeah. And so that's how you can tell that it's a lizard, if you can get that close.
Additionally, you can see on this one, it has eyelids. Its eyes are closed right now.
Uh-huh.
And that's also something that lizards have. And then underneath, I'll do it again. Underneath, it has small little scales that look just like the scales on top of the lizard. Whereas on a snake, let me grab the rubber boa again. On a snake, the scales underneath are these long scales.
Right.
That cover the whole belly, basically. So if you go back to the slide, we have kind of a zoomed in version in case it was hard to see on the specimen. But so these ventral scales on most snakes, you'll see that they have these special scales on the underside that are different from the scales that are surrounding the rest of the body.
Mm-hmm.
So it does require getting a little bit intimate with the animal to be super confident if it's a lizard or a snake until you get used to your local diversity. But those are some key giveaways. It's not just the presence or absence of limbs.
Mm-hmm. Its face was really incredible too. Yeah.
And again, like Lauren said, like you just kind of start to get used to like, oh, that's a lizard looking face.
Right.
Or that's like a snake looking face.
Right. Very cool.
All right. Who's next?
All right. So yeah, Lauren alluded a little bit to the 1905-1906 Galapagos expedition, which resulted in a really significant collection for herpetology, but also for basically every department of the institution. And it was made even more significant or important because of the events in 1906 when there was a major earthquake in San Francisco and when the academy at the time was mostly destroyed by a fire after the earthquake. And we lost most of the scientific collections as a result of that. And so this expedition, which was already meant to really bolster the scientific importance of the California Academy of Sciences, ended up just being like the most important expedition that the Academy had ever planned because it reinvigorated the Academy and basically saved the Academy.
And so they knew when they went out on the expedition to the Galapagos that a lot of the species there were in peril. So they tried to do a really systematic kind of overview and look at all the different islands and collect from all the different islands because each island has different species. So that noise is our frog clock.
Of course it is.
Every hour there's a different frog call. So ignore it, but here in California, it's 11 o'clock. So they went to different islands, collected things across the archipelago, and it really was like the first systematic kind of survey of those islands. But
yeah, it provided just sort of a baseline of like what's here, which species are there, which islands do those species occur on, and it was really the foundation for everything that we've learned about the Galapagos since. Because even though, of course, Darwin made a very important expedition to the Galapagos, he didn't do this type of really systematic surveying And so a lot of the research that has been conducted since then and our understanding of all the amazing species that live in the Galapagos is based on the California Academy of Sciences collections and other similar collections that were made after Darwin.
Mm-hmm.
So I'm not showing our tortoise room, which is very impressive, but back then they preserved the tortoises with arsenic, which is a carcinogen. It's very toxic. But we also have a pretty extensive skeletal collection. And so I'm just showing off one skull from one of the tortoises. This is from Pinzon Island, and it was collected in 1906. What we're trying to do now, because these collections are relatively inaccessible, not only because of the arsenic, but because they're fragile, they're huge, they're old, and they're really historically important. We are now digitizing all of our collections, and so we're planning to use CT scanning to create 3D models of the inside and outside of some specimens, and then also use 3D surface scanning to create 3D models of a lot of the tortoises and things that wouldn't fit in a traditional CT scanner, like a medical CT scanner. And all of that information will be available to the public. You'll be able to search online for these 3D images and see more of the collection. And those images will be like scientifically relevant. They'll be to scale, and so people can do measurements and morphological studies with them.
And if you want to see our current 3D collection of specimens, if you go to Sketchfab and search California Academy of Sciences, we have about 700 there. And I think there's a few sea turtle and tortoise skulls in there potentially. So I'll drop a link to that as well.
Yeah.
And just to be clear, because somebody asked, they just wanted to know, are you talking about the really, really big, famous tortoises, slow-moving, long-lived tortoises that everybody's kind of familiar with? Okay.
Yeah, huge, huge tortoises.
Yeah, and so one of the things that is really unique about the collection, and that also is unique about this digitization initiative that Lauren's talking about, is that we've known for a long time that evolution relies on diversity in natural populations, and that natural selection is acting on that diversity where certain individuals are going to be more successful and leave more offspring to the next generation. And so because of that, we don't just want to have one individual to represent the species or even the population, because we know that diversity in that population is what evolution uses basically for natural selection. And so the digitization effort includes scanning more than just one individual per species or one individual per island to really have this representative subset of all the diversity that exists within a population when possible. And that makes it more relevant for scientific research, but also more relevant for educators when they want to teach concepts of natural selection in their classrooms.
Right. That was
and
really
so there'll also be lesson plans associated with it. Yeah, and it's not just the herpetology collection that's part of this initiative. There are also birds and mammals that will be scanned as well. So we're really excited about the potential of bringing this resource to a wider audience, because like Lauren said, there are a lot of logistical reasons why we can't necessarily send these items out on loan because they're so precious and also really bulky. And then of course the ones that are covered in arsenic. So there are just all these challenges for making them accessible to researchers and the public. And so this digitization initiative is hopefully going to change some of that.
That's really exciting. Is there a timeline for when we might start to see the result of that?
The project starts next month. So okay. Things won't happen fast because we're still closed, but the project will run for the next 2 years.
That's great. Great. Really cool.
Okay. All right. So next we have another, hopefully almost as iconic as Galapagos tortoises, thanks to the internet, but a very famous frog that you may have seen. I forget when this was going viral, a couple years ago. But so these are desert rain frogs, and they live in the deserts of southern Africa. And there's a bunch of different species, and they all have, again, this kind of like similar funny looking body plan as that Mexican burrowing toad that we showed you. And that's because they spend a lot of their lives underground. And this little species has an adorable battle cry that we'll play for you. And then Lauren will show you the real deal.
I was trying to laugh quietly.
And I think this will be made even more adorable once Lauren shows you how tiny these little frogs are.
So mad.
So this
So
is
angry.
an exact species. There are a lot of different species that look pretty much the same as this little sandy desert frog, but this is a different one, and you can see
It's too much.
they do tend to have this same body plan, which is, I think, good for the dry environment. But yeah, they all have, I keep going the wrong way, these really grumpy little.
It's perfect. It's clearly the most perfect animal today.
Yeah, it's hard to pick a favorite, like we said.
Yeah. I like how its battle cry really kind of rocks its entire body. Like it takes a lot of work. And when does it actually make that sound?
So I think in this case, it was feeling a little bit threatened just because there was a human there that had startled it as it came above ground. I saw one for the first time. I was in South Africa late last year for a conference, and I went for a hike and got really lucky and saw one, and it did not squeal at me. So they don't do it every time that they see a human necessarily, but I think this one was feeling threatened.
Maybe they had a meeting and they were like, for a long time we thought this noise really, really scared humans.
Right.
We're starting to think it's not that intimidating. That's incredible.
That's true, because my experience was post-internet sensation.
Yeah.
So maybe the word is out.
Gather more data.
All right. So I think this is our last little anecdote. And I'm going to let Lauren take it away on this one. This
is a weird one. It's not really that specimen based. It's mainly about a historical character that used to be associated with herpetology. So this is Joseph Cheeseman Thompson, also known as J.C. Thompson, or Joseph the Snake Thompson. He was at some point a curator of herpetology at the Academy, but that was after a career as a naval surgeon, as a spy. So he did a lot of spy work in parts of Asia, and when he did, he had an alias, which was Victor Kuhn. So this other picture is him dressed as a herpetologist, a Belgian herpetologist named Victor Kuhn. As both J.C. Thompson and as Victor Kuhn, he collected about 12,000 specimens for the Academy's collection, and that's just for herpetology. I think he also collected something like 30,000 insect specimens. But also, in addition to being a spy named Victor Kuhn and J.C. Thompson, he wrote books on Buddhism. He was an early influencer of L. Ron Hubbard and a big character in Scientology.
Oh, wow.
He was one of the first people to bring Burmese cats to the U.S. and became a noted cat fancier. So he's kind of an enigma. He's a weird part of art history at the Academy, and I like that his things are around. So I brought 2 specimens. They're not that exciting, but what is kind of fun is they were both collected at the same time in the same place in Okinawa, but under both names.
Oh.
So about 85% of what he collected is under his alias, but some things are in the catalog as J.C. Thompson. But yeah, just an interesting character I thought I would bring up.
Yeah, he's like too productive. Like,
Yeah.
calm it down. That's too much. Also, it seems like not great spycraft to collect under both names in the same place at the same time. Like that seems like a misstep to me. I mean, I don't know. Also, this is why people don't trust scientists. You like go into foreign countries and you're like, I have business here. Like I am doing things. It's all his fault.
Yeah. No, he was kind of a weirdo, and he was a contentious man. I think he had a lot of issues with the existing curators. There were stories written about, battles over naming specimens, and he was a character.
Like where did he find the time to get into battles about that? It's just like between my Burmese cats and my collecting under multiple names and whatever, I'm gonna have like a rigorous argument with the other staff at this institution. Yeah, it's great history. Yeah, excellent.
So that's all of our specimens that we have out.
Okay, that was a lot. People loved it. Thank you so much. I know it was like took some work to prep all of that stuff. Let's see. I'll ask just a handful of questions, and folks, this is your last chance to get them in. So this one from Kristen again, I'm just going to ask this question because I don't know anything about this. Is it true that frogs or toads are found entombed inside of rocks? And if so, how does it happen? Does this make sense to you?
Wait, can you repeat that?
Entombed inside of rocks?
Maybe I won't. Okay. He asked if it was true that frogs and toads are found entombed inside of rocks sometimes. But I don't know if that could be like a fossil amber question. Maybe we don't know the answer to that.
I don't know the answer to that.
No. I mean, sometimes they can be entombed in ice and dirt, and then they can kind of come back to life, even though they were alive the whole time. But you can find kind of an icicle of a frog up north, and they can come out of this frozen state. But I haven't heard about real rocks.
Maybe that's what they meant. And so that's like a hibernation. Is that a type of hibernation, a seasonal thing?
Yeah, torpor, yeah.
Okay, great. So let's see here. YouTube user Darthver1. I think this question was about the Principe tree frog, but asked, are those pads using the van der Waals interaction with gecko wood?
No. But that's a really good question. Yeah. So the sort of microstructures of the toe pads of tree frogs are not as sort of like powerful as geckos and anolis lizards in that sense, but they are still very good at climbing.
Okay. And how did those work sticky wise?
I think it's just kind of like a suction cup, like surface area, but it's not like a microstructure chemical bond type of situation the same way that it is for geckos.
I see. Okay. Thank you. Seth had a question. Is formalin part of a good skincare regime? I think the actual question is, does it hurt your hands to handle these things, or are there any other considerations in working with collection?
No, so formalin is not a good chemical. Formalin's carcinogen, it's a mutagen, and generally, no, like you don't want to be anywhere near formalin. We preserve the specimens in formalin, and it gets absorbed into their cells, and then all of the excess formalin becomes a gas and gases out. And so eventually, we soak them after they're in formalin, and then all of that excess formalin that's not absorbed should be gone when we put them in alcohol. And so I can touch them. Researchers, when they use the specimens, can touch them. And right now my hands just smell like slightly snaky alcohol hand sanitizer.
Wow.
Yeah, there's some specimens that stay in formalin. Tadpoles usually will stay in formalin even after they're preserved. But for the most part, it's just a temporary exposure so that they get really stiff, like one of our previous viewers mentioned that she noticed. And then once it sort of stiffened the body of the specimen, it's done its job, and it doesn't need to remain in formalin to retain that stiffness.
Okay.
So we just transfer it to a less toxic.
Mhm.
And prior to about 1950, 60, they only preserved in alcohol. So they didn't even use formalin. So that's kind of become a newer thing. But with the older specimens, there was no formalin involved.
Okay, great.
And just as a side note, I do think that formalin or formaldehyde is used as a chemical hair relaxing treatment. So I don't think it's used in any kind of skincare, but I think it is used to straighten hair.
Yeah, I feel like it might be present in several iterations in like the beauty world.
Yeah, like probably not something that you want to put on your body.
Right. Yeah, exactly. Okay, I'm going to take this one from Miranda, so basically she's a college student who wants to be a herpetology curator specifically, and asks if right now, as a college student, there's anything she should be doing beyond basic biology classes.
So for sure, take a herpetology class if you have one at your university.
Yeah.
Not all universities have them, but definitely great to take one if you can. And then I would recommend reaching out to herpetologists at your university or at other local universities if your university doesn't have a herpetologist, or a natural history museum that is somewhat nearby. Often there are opportunities to volunteer. And so it's always a good idea to reach out. And I think especially if you know your stuff and have already done a lot of sort of preparation ahead of time to show that you're serious, then people will be excited to give you an opportunity to contribute and learn more and see if it really is something that you want to commit to working towards.
Right. And we didn't get this question today, but we often get asked by undergraduates and grad students what specific kind of skills or techniques, whether it's related to genetics or software or whatever else are, and I would say for collections management as well, are there any specific kind of skills or areas that you think are particularly important now and moving forward that people earlier in their career should be thinking about or trying to get access to?
I mean, I think I'll just speak to research, and then Lauren can talk about the more collection side, but for research, one of the things that I really like about being a herpetologist or being an organismal biologist is that there are tons of different techniques and research questions that you can pursue. And so there are just a lot of different ways to be a herpetologist. And so you can be really interested in evolution, in which case you probably do want to learn genetic or genomic research techniques. You might be really interested in ecology and behavior, in which case you might want to get really good at statistics, which might seem surprising, but statistics end up being really important, and getting experience working with animals, just learning how to set up these types of experiments, and sort of the tediousness of trying to quantify behavior. Mm-hmm. And then some people are really interested in disease and conservation. And so that can end up being a combination of ecology and evolution. And then other people are really interested in morphology and functional morphology. So like how different structures are involved in things like capturing prey, or other sort of like sprint speed or gripping ability, some of the things that we talked about a little bit today. And so often those people need to be really good at the anatomy and morphology. So getting experience, even if it's not a herpetology or vertebrate anatomy course, getting experience in just general anatomy will be really important. And then you might want to get experience with skills like CT scanning and physics. If you're interested in biomechanics, you'll probably have to have some physics. So it's really like, obviously you can't be an expert in all of those things, but there's so many different ways that you could bring a unique skill set to better understand herpetology and the diversity and evolution of all these really cool organisms.
That's so cool. And it sounds like the first step is really just trying to figure out what you're genuinely going to be interested in for the next 40 years or something, like what area.
But it can also change. Like I said, I'm studying vision in frogs right now. I could never have predicted that as an undergraduate that I'd be studying this. And honestly, even in graduate school and during my postdoc, I never could have guessed that I would be studying this right now. So you also can continue to grow and learn new skills as you go. And you can also collaborate with people that have skills that you don't have. As long as you have something to bring to the table, people will usually be excited to collaborate with you and sort of expand the types of research that you can do. So I think if you're really interested in being involved in a natural history museum, I think the one unique thing that you really would need to get experience with is working with museum collections and using museum collections in your research. So there are a lot of herpetologists who do research that isn't based on the voucher specimens like Lauren has showed you today. And natural history museums are kind of supposed to be this like special place where that is a really big part of our research program and where we see the value in having this resource and also are thinking of innovative new ways that we can use this amazing resource to study new questions.
That's great. That's great. And Lauren, what about for people interested in, I mean, I imagine for a lot of people watching today, they didn't even know collections management is a role. So what might be useful for someone interested in that direction?
I came at it in a kind of different direction, whereas I did both an undergrad and master's degree in biology, in evolution and ecology, but not specifically in herpetology. But my first job after undergrad was in a science museum. And I have worked in science museums consistently from that time. And so I've been working at this museum in various positions since 2008 or so. And I started here moving the collections as a temporary job. And then I volunteered in birds and mammals. I worked a little bit in entomology, and I was a research assistant for a grant in herpetology for 2 years, which then led me to working as a curatorial assistant in the collections, where I worked for many years until kind of graduating to my current position about 3 years ago. So my actual academic background wasn't in herpetology, but I've had a lot of on-the-job training in the last 10, 12 years.
Yeah. It's always great to hear about people's kind of journeys too, because I love that there's not one path. So yeah.
But it definitely came from a love of museums. Like I love museums. I like things. I like organizing things. My job is kind of anal organizer, and I do like that.
Does your closet reflect it? Rain is not.
I'm like a little bit of a bear in the house, and I'll like throw things around, but my closet is like, it's organized.
Okay, good. I'm glad to hear that. Yeah. So I'll just end with this last question because I thought it was a nice one. It's from Anna, and she asked it when you were talking about, when both of you were talking about the Galapagos expedition, and she asks, have collections techniques changed since the early expeditions? And I guess I would add techniques and philosophies.
Yeah, so for sure the techniques have changed. And I would say that the philosophy, certainly in what Lauren and I have learned about the motivation for that Galapagos expedition, I think it is very much in line with our philosophy today, which is that there's a ton that we don't know about the biodiversity on this planet, including biodiversity in California, and that we know that it's at risk. Like that's the one thing that we do know is that it's at risk, and we can't effectively manage that risk if we don't understand at a very basic level the centers of biodiversity. So just how many species are there, where do they occur, and then some very basic things about their biology. Because we just can't make management policies that will be effective if we don't know what we're protecting and we don't know what they need. And so that was very much the motivation for this expedition to the Galapagos, where there was a lot of popular writing at the time, especially about the tortoises, because tortoises had been extirpated or, hunted to extinction on lots of other small islands around the world. And so the scientific community was seeing that same trend in the Galapagos, and they were really concerned that everything was going to be overhunted to extinction before we even knew what was there. And so I think that same philosophy is true in many parts of the world today, not just held at the Academy, but lots of other researchers and nonprofits and government agencies that are really concerned about just documenting the biodiversity that we have. So that is certainly still a very similar philosophy. In terms of the techniques, of course, we have so much technology available to us now that they didn't have at the time. And I think one of the most challenging things about being a museum-based researcher is that we have our own research interests, and we're picking places in the world that we have a scientific question that we want to address, whether it be in Northern California or the islands of Sao Tome and Principe, where Lauren and I have done work. But we also are trying to anticipate the needs of future generations of scientists and future societal needs. And so we're basically trying, just like our predecessors were 100 years ago, to kind of get this baseline data and baseline survey of diversity so that for the next century, people could start to document species diversity and study the ecology and evolution of these species. We're kind of trying to do that as well in the places that we work. And so it's stressful because we're trying to anticipate the needs of future people and future technology. And so a lot of these historical collections that were made over 100 years ago, they had no idea that we'd be able to CT scan specimens or sequence genomes from tiny fragments of preserved tissue, to do stable isotope analyses, to look at the diets of different organisms. So just these amazing technologies. They didn't know about all these climate models that we have, that we're using the location information from a specimen to try to understand the climates that that species can occur in, and where those climates might occur in the future, so that we can preserve not only current habitat, but future habitat for species. There's just like all this amazing technology that we're able to use with the data that people collected 100 years ago that they never could have anticipated. Yeah. And so it like really does keep me up at night of like, did I collect enough data, and did I like adequately think about what future people might want to do with the specimens that I'm contributing to the Cal Academy's collection? And I know Lauren feels the same way. It's just like, did I get every possible thing that I possibly could? Not just for me, for my research, but for all the people that are going to come in the future. And also thinking about like some of these places, which fortunately hasn't happened to me yet, but I know it's happened to colleagues, where you go to a place and you find this amazing biodiversity and you try to document it as well as you can. And then that place might be gone in the future, either due to something like a catastrophic fire or a mine, or, you know, just there's so much going on in the world. And yeah, it's stressful.
Yeah. Each specimen that we have really is like a snapshot of both like in time and in space.
Yeah.
So we really need them as a historical record.
Yeah. And right. Yeah. And just like, so all the stress of trying to make sure that you're doing thoroughly and anticipate all of that stuff. And then also just racing these huge things that feel unstoppable sometimes, like climate change and habitat loss and everything else. Yeah. But I mean, the fact that part of your profession really means thinking about all the people who will come after and continue to do the work is really inspiring. And yeah, it's really, I think you both said it so well. Thank you. Thank you so much for being here today. And again, for all the work it took to put together the tour itself, the virtual tour, which people loved and already want more of. So I don't know, maybe you should like figure out how to do like collections within collections. And Raina, I know we'll get you back next month to hear more about Sao Tome and Principe. And Breakfast Club viewers, on next Tuesday, if you come back here at the same time, we will have Dr. Jack Dumbacher, who is our curator of birds and mammals, and he will be talking about the poisonous birds of Guinea, which you didn't know existed, but are about to find out about. And before we say goodbye, I will just say that as the Academy prepares to enter our 6th month now of closure, We really do need your help more than ever. And we know things are incredibly hard for everybody. So if your help just means continuing to watch and engage with programs like this, that's wonderful. If you are in a place where you can comfortably give, we would welcome donations of any size. We are continuing to care for our 40,000 living collections, live animals, and support as many staff as we can and protect as many jobs and ensure that we're here to reopen as soon as we can. So if you can give any amount at all, we have a donate button on Facebook and a donate link on YouTube, and we thank you so much for that. So again, Lauren and Raina, thank you so much. That was so cool. And viewers, thanks for being here, and we will see you next week. Bye.
Stay safe, everybody.